Hello, and welcome to the inaugural episode of Curate This, a podcast brought to you by the International Society for Biocuration, where we're going to be pulling back the veil and showing how ontologies, databases, and other curated resources are made. I'm Charlie Hoyt, and I'm coming to you from the future with a minor announcement that we had some audio issues when recording this episode. Hopefully, we'll iron out the kinks in future episodes as we get more experience doing this. Today, we're talking to Dr. Susan Bello, who works as a curator for the Mouse Genome Informatics Knowledge Base and the Alliance of Genome Resources. Sue's located at Jackson Lab in Maine in the United States. And for MGI, Sue primarily focuses on curation of mutant alleles and the phenotypes associated with those alleles. Sue is also the chair of the executive committee of the International Society for Biocuration, so we figured she'd be the perfect person to start with. So the format for this, we're going to talk a little bit about MGI, and then we're going to look into how some curation is done. And Sue's going to screen share and walk us through how she works on a normal day. So we've got a couple questions that we're going to try asking about all of our resources. The first one we've already mentioned is that the resource Sue works on is called MGI. First question is, how old is this resource, Sue?
So I had to go look that up because I can't always remember. It was first formed, MGI 1.0 came out in 1994.
Wow.
Sorry, technically that was MGD 1.0. So MGI is actually not a single resource. It's a collection of resources that we've grown more. But MGD, so that 94 date is the date it was first put out on the internet as its 1.0 version. Before that, there were a number of other resources going all the way back to the 80s when they would send out the mouse locus catalog on floppy disks so that you could get your own. Yeah. So
I was born in 93, by the way. So next question is MGI, is it developed as an isolated resource, or how does the community interact with it? Is this open to external contributions? Do you have partners outside of Jackson Labs?
So yeah, I was looking at that question and thinking, what does isolated mean? In terms of actually annotating into the database, you have to be an MGI curator. We don't have a lot of community curation. We do have the ability for the community to send us input. We have submission forms where they can send things. We do a lot of data loads. There's a lot of interaction with the community. So, I looked at the isolated and I'm like, no. And yes. So yeah, we don't I know some groups have actual researchers, community members will curate their own data and that will be populated in the database. And we don't really have that sort of direct. I think primarily because the mouse community is so large that it's harder to get that sort of buy-in. So yes and no.
Okay. So how many people are working on MGI or within your group?
Yeah. And I had to go look that up, and the staff is over 30. If I include all the PIs, I think it's either 33 or 34. It's smaller than it used to be. There used to be more of us, but we have sort of trimmed down over the years as our budgets have gotten trimmed down. It's never enough people.
Okay. And then the big question is what kind of information does MGI store? And does it develop data standards for that kind of information or participate in any community efforts for doing data standardization?
So MGI is essentially everything about mouse genomics. So it's got the stuff I work on, which is phenotypes and alleles and disease models. We have developmental expression. We're part of the GO group. So yes, to collaborating with external groups, because the Gene Ontology Consortium is a consortium that is spread around the world. Yes, to developing standards. So we're the authority for mouse nomenclature, but then we collaborate with the mouse, human, rat, have a nomenclature committee so that we can have unified standards for nomenclature. We develop the mouse phenotype ontology I did it. Mammalian phenotype ontology, which we use for describing mouse data, is also used by the rat genome database. So, and it's designed to be used by any mammal. So that's another thing I work on. Then we work on the gene ontology. We have a mouse anatomy. We have development. So yes to lots of standards.
Okay. So next big question is, what is the downstream impact of MGI and the work that you've done and the work that your colleagues do, and who uses these resources and why?
So the resources freely available to anyone and everyone in the world who wants to know anything about how mouse genes work, what are the impacts of a mutation, what models are there out there for modeling your, if you're studying a particular human disease, what models are out there, you can easily come search. I think the like elevator pitch I usually give for what I do is, if you want to know what genes affect heart development, you can come and quickly do a search and find that. In terms of users, I mean, we have pharma companies that just come suck the entire database up and go do something. It's some of the more interesting things. It's been incorporated into some variant predictor tools for when you have an undiagnosed disease patient and you've done sequencing and you then have variants that you're like, well, maybe it's one of these. Can I use a variant predictor tool? They can incorporate all of the mouse phenotype data, the mutation data in there to say, oh, this is probably the variant I want to look at. And this is, there are cases where that has then been used to help justify a course of treatment for people. And I think that's one of the more interesting, sort of really impactful things. It's nice to see.
So more or less, MGI's resources are used worldwide by pharma
Yes.
companies in translational medicine and also in the clinic.
And in the clinic and in basic research. And
Do you have any way of counting, what a quantitative way of saying what's the impact of MGI throughout this whole ecosystem of researchers and clinicians and practitioners?
so the PIs, like there's the basic of how many people use us. So they look at, the hit counts, but that's hard. So the answer is yes and no, and I don't have that easily to hand.
I think in general in our community, we have a very difficult time assessing the impact of our
Yeah,
works outside of literature citations, especially because it's difficult to really know which things are used indirectly.
yeah, and it's hard to know because people don't necessarily cite that they're using you. They just come and do their background research for some project. And so they've used you, but you don't necessarily get a citation. And then with pharma, they come and they just take the data and we don't know really what they're doing, which is fine because they're using the data, but you don't necessarily get to quantify the final downstream impact.
I think we're ready to transition into the second segment of this interview, which is actually to show us you doing some curation. So before we get into that, can you explain what kind of curation you're going to show us? And then we
Okay.
can get into it.
So yeah, since we were trying to keep this short, and I decided that we would just sort of focus at the new allele entry. So MGI, one thing MGI does is goes out and gets basically every paper in PubMed Central, go out to the API, and if it says the word mice in that paper and it's not in the references, we pull it in. And then we have an algorithm that like runs through and scores things. And then curators come in and say, yes, we want it. And, one of the things we say is, oh, this has a new allele in it. We need to create an entity, a new allele entity for that and document it so that we can attach all of the data to it.
Can I ask a quick question just for
Yep.
people who aren't aware? What is an allele?
So an allele, and you would not believe how long we spent defining this word at the Alliance. It is a specific set of variants for a specific sequence. So you have a gene and it has, you sort of have like, okay, this is the gene. And if you have these three changes, this is this specific allele. And if you have these other changes, it's this specific allele. So we've defined a variant as a specific contiguous change in the genome. And we defined an allele as a collection of one or more changes to a specific region in the genome. And I'm using very wobbly words because I don't want to say gene, because you can also have alleles of like, regulatory regions that maybe regulate a bunch of genes, but you've made a change. And so now we've got to capture that.
Okay, so what we're going to do is Sue's going to screen share and walk us through the curation system from MGI. And whenever you're ready, you can do that. You can voice over and narrate what you're doing
Yep.
and as you're doing it. And I'm going to sit back and enjoy. Maybe I ask a question as we go, but otherwise I'll keep quiet.
Definitely ask questions as we go because otherwise this is going to be a little tedious. And I'm probably going to use jargon that is MGI specific. So I will just say this at MGI, we refer to this as the EI because it's the editorial interface. And so before this started, I actually did a search, which was looking for my name and a paper that had a tag that was for new alleles and didn't have a tag that said it was we actually have to talk to the author, or it had some problem, or it was incomplete. These papers have all been tagged in this way to make it easier to do this.
Is this all automated tagging, or this is like you've gone through and
We've
already
gone through
triaged a
and
little bit?
tagged. So
Okay.
the priority tag is automated, and that comes from now you get to enjoy screens.
So wait, so you clicked that search with the tags, and now we've got a list of papers.
Yes.
Okay.
So this is my depressing backlog of 885 papers that have new alleles in them that I need to enter. And so I picked one and I did a lot of pre-screening because I wanted one that was actually nice and that we could get done and yet still had most of the steps in it.
Do you think you can make the text a little bit bigger? I'm not sure how this is going to look when we're looking at the screen share later. All right, so just walk us through what are all the fields on this form right here, because there's a lot going on.
This like big section here is basic paper information. It's what you would see in PubMed. You get the PubMed and the DOI, and then the authors and the title, and then there's a box for the abstract, and then we have internal notes that nobody else sees. And in this case, it says, "Oh, there's an existing allele, but there's also a new allele." And then here's all the tags, and then we have a way to add tags. And then we have, because MGI is a bunch of different projects, each of the projects keeps tracks of whether or not they want the paper. So AP is alleles and phenotypes, and we want the paper. And I went in ahead of time and indexed it to say, when I opened the paper, yeah, so this is complicated. So this is the basic interface. And then down below, it keeps track of like all of the history, and it has relevance. So it has the lit triage load, which is the automated thing. And then it has a classifier, which gave it a score. If the score is below a certain level or it's negative, it just gets put in the discard pile and we probably will never look at it. But then two of us have come through and said, yeah, we definitely want to keep this. And then I went through ahead of time, having opened the paper, and added the existing allele that was in the paper. So, and then there's all sorts of other things you can attach to the paper directly here so that you don't have to go to a separate screen.
Okay, so when you look for this existing one, this you reference against the MGI's database already to
Yes.
find like there's some text in the paper that mentions this allele, and we
Yep.
already have an entry on that. And so you're
Yeah.
just like saying in this curation interface, hey, we've already done that. That's old news.
Yeah, that's old. That's good. And so this paper is now attached to that. And when this makes its way to public, it will show up in the reference list for that other allele.
Okay, but we're
Here's the
here
paper.
for the new and cool stuff.
We're
So
here for
show
the
us
new and
how
cool stuff. So there's
you
a
find
handy
the
little
new one.
button that lets you open the PDF right here so that you get the PDF immediately. Yeah, and I tried to make things pretty big. So
Yeah, I think this looks pretty good.
okay. All right. And from here, then there's a number of things we have to figure out. So I'm going to say, all right, here's the paper. I'm going to hop you over to another editorial module, which is for creating a new allele. And again, there's a lot of things here. There's a lot that we have to attach. Not everything gets attached to every allele, but so I have an allele module. The first thing I've got to figure out is what gene are they talking about? Because they're calling it TWIC1. Is that actually the gene nomenclature, or is that some other gene? So for that, I would just, I do a lot of copy paste. So I just
So hold on, can I stop you there? But can you go back to the PDF? So you start by looking at the title, and the title is saying, okay, we're looking at mice that have some change in this gene TWIC1, and most of the literature is pretty like focused this way that you end up looking at, or sometimes you have to go into the abstract or like read through
I
the
have to
methods section.
go all through the whole paper. Yes, I skipped way ahead.
Okay, so this one is like a pretty easy scenario where the title already gives you some hints as to what gene.
The title, and honestly, it was really easy because they gave you the two alleles in their graphical abstract.
Nice.
They're like, okay, well, this is great. There's an exon 2 deleted and an exon 1 deleted. And usually the first thing I would then do is just hop straight to the methods. Like I ignore the rest of it. I go to the methods and I say, okay, you've told me that there are these two alleles from your graphical interface. All right, I can now see, okay, you've got two alleles for this gene listed here. One of them is from this other reference, and so I went and looked that up and added it. And this one says it's this paper. Okay. So then I go and say, okay, this paper, animals, and then they give me a description, right? And again, they're telling me this allele was gifted from that person and this allele was created here. So this is how I know absolutely this allele was created here.
Okay.
Then I do still need to figure out what gene it is. Because sometimes you stumble at that block. They give you some synonym and you can't match it to a single gene, and that can take you a while. But so that's usually the first thing I go look up, because that will also then let me answer other questions. So I can see that TWIC1 is actually KCNK1.
Why do you think that they use TWIC1 if the official symbol is KCNK?
Probably because that's what their lab has always called it. Or that's
like their lab is like 40 years old before stuff got standardized.
Or that's the branch of like their community has always called it that. And so that's what they're going to keep calling it.
Okay.
Even though officially it's been named this. It is what it is.
All right. So what kind of information, now that we've kind of got an idea that we know which gene it is, we found information about the allele, and I saw that there was like a sequence that went along with it. What
Yep.
are we going to put into the curation interface now?
Well, the first thing I'm going to put in is this gene symbol, and I'm going to do a search because yes, they say they created it, but some journals require that you have an ID. This one clearly didn't. And some groups are good about reserving their allele symbol and reserving everything ahead of time. Generally, the first
Do they
thing
reserve it with you?
Yes,
with
they
MGI?
reserve it with us.
So they have a different curation interface for that.
We have a form which says submit data.
Mhm.
And on the submit data form, you can submit your new allele. And so they would send that to us and we would work with them to get it all set.
Okay.
But this group, if I go back
They
to
didn't
the
do it.
they didn't do that. And and it may very well be that if they had done that, it would have been a different MGI curator who handled it. So the first thing is always check, is there something reserved? No, everything. So I can see, the very first thing here is approved. The handy thing is there is nothing private with this one, so I didn't have to worry about screen sharing. So we have when I run this search, I get the full list of alleles over here, and they're sorted by status first. So if I'm seeing approved first, then I know that there's nothing that's not approved. Because if it was reserved, it would have been it would have been before the approved.
So like it's something that's embargoed that a group hasn't made public yet, or maybe it's proprietary.
Usually if a group is reserving before publication, they often ask not to have that released until it's published. And so that's there. There's also sometimes deleted things, but yeah, we will keep anything that you request be confidential until you publish it.
Okay. So this list lets you know that it's not in the database already. And next step is to start filling in the form.
Yep. So go back. We say this is one of the things about the way both mouse and rat name alleles is the way we name an allele depends on how the allele was generated. So you go and say, it's a CRISPR allele, which honestly these days, if it's not a CRISPR allele, I'm a little surprised. Most of the new alleles are CRISPR these days. Everybody really likes CRISPR. So if I come in here, I can be somewhat lazy and click on an existing CRISPR allele.
How do you know it's an existing CRISPR allele?
Yes. So nomenclature for mouse and rat for alleles depends on the method of generation. So anything that is CRISPR or TALEN or zinc finger nuclease is an endonuclease mediated allele, and their superscripted portion will start with E, and the allele name will start with endonuclease mediated. So
you're going to choose something like 1CSC, but you're going to make up one of these now, or is that systematic?
So I deselect it so that I now have a clear screen, but it held on to like the symbol and the name and some basic information. So, all right. So I've got sort of a starting place. The next thing is when we create an allele, the letter codes at the end is actually a lab code, which says this person is responsible for the creation of this allele. So we have to go back to the paper to figure out who this person is.
So you're taking the corresponding author who's like in charge of the lab of the first author?
So usually what we'll do is we'll look at, all right, so there happen to be in this paper 2 corresponding authors.
And one of them
then,
corresponds to the first author. So you
yeah,
pick that one.
actually, in this case, no,
Okay.
because then you have to check resource availability. I also usually check the methods for how it was generated. So if I go to the methods, right, the methods that we looked at initially just said that these were generated by CRISPR through customized targeting service. So it's not that they bought it from a company, because that's usually the first thing is, did they just order this from a company and the company will sell it to anybody? That's not the case here. It's a customized thing. The author most likely has the responsibility. So then you go look at the author contributions and contact information. And in this case, they gave me a very nice statement, which is specific requests about this TWIC1 exon 1 targeted, which is our new allele, should be directed to this person. Super helpful. I know precisely which person all the requests should be going to. That's the person I'm going to use. So in fact, it's not.
So we're doing JYP as the
We're doing this
acronym.
person. And even though, yeah, as you initially thought, oh, well, this lab code corresponds to that. That's not the case in this one.
All right.
We're going to go with this guy. Then I need to go. So MGI and RGD both use the ILAR lab codes, which is run by the national academies. So it's external to MGI, and there's a database of all the different labs. And so I would go and I would say, all right, last name is Park. Let me search. Okay.
Oh no, we're going to get a million.
Boy, there's a bunch of Parks. So then I use the control F and I just search for like part of their name to see, is it there? And in this case, no, neither of these parks match the park that we want. So I know I need to make a new lab code. Then I have to say, okay, well.
Wait, so we're going 2 levels deep? Like now we have to curate another database before we can curate your database.
Yes.
I love it.
I got to make another I got it. So by convention, we usually try to start with the first 3 letters of their last name. So I just look for PAR to see what's available and what's already taken. And in this case, what I found was that, okay, there's a bunch of PARs, but there's no PAR J. Oh, first 3 letters of your last name.
Wait, well, hold on. I saw there was a Park.
There's a Park J, yes.
Yeah. Okay.
But there's no PAR J. So that's fine. Doesn't matter to me that there's a Park J.
Okay.
I need 4 letters that don't match. You know, I don't know. So PAR J works. Then I go and I Google PAR J, or in this case, I DuckDuckGo PAR J.
Uh-huh.
I need to
make sure that we're not using some swears in other
I don't
languages.
Want to use, yes. I don't want to use a slur. I don't want to use like anything that could have a negative connotation in some other language. And so I can see, all right, yeah, it's a boy's name. That's fine. I don't care about that. I care that it's not going to be offensive. So we've learned over the years that you check that. The other thing you have to check is that it's not already in the system as like a gene name or gene synonym, because you don't want to have like this confusion. So in this case, we're good. It doesn't have a negative meaning. It's not in use. It doesn't match up to anything in the system, which is great. So then at the ILAR site, there's a form that you get to fill out, which says, all right, here's the and I pre-filled this because it's a lot of typing. Form, fill in the name, fill in where they are, grab all the information about where they're located. And the key thing that we have to fill this in, you can skip a lot of the fields, which is great. You have to have an email.
Mm-hmm.
That's why it's always been the corresponding author that we like submit these to. And then I will just submit this and we will have our lab code. So it gives you a little thing to double check. And then you say, okay, and off it goes. And I now have a little message that says the code has been successfully created.
Woohoo, curation. We did it.
We are good to go. So now I can just come in here and I can change that. And then I can change this to our author's name. And I'm going to copy it because I might misspell their first name. All right. And we just make sure there's no weird first character. I usually, any dash that's in there, I get rid of and put back in because I don't trust the formatting.
Smart.
So then I put this in. All right. So we got our first step. We've got an allele symbol with our new lab code. We've got an allele name. Got the gene. We're going to go back over here and we're going to copy the J number. Every reference that's in MGI gets this identifier because this dates back from when we actually had filing cases full of actual paper to work from.
Wild.
Yes. And so they are the J files. And so every paper has a J number. It's just one of those things. Every allele must have an original reference. That is the one reference it is required to have. And then it also gets a molecular reference in this case. And that is the reference that gives you the description of the molecular mutation. And sometimes for like an ENU or some of the spontaneous ones, the original and the molecular will be different. But most of the time for the newer ones, they're the same. So then I can flip the status at this point to in progress, just to be safe. You pick the generation method. So like I said, the name is dependent on the generation method. And then we also specify, because sometimes if like you've used CRISPR or targeting to create a really big deletion, then the name might not have this particular text in it, but we'll specify what the generation method is. And this is just a drop-down list. And so you just pick so that you can't mess that up. For inheritance mode, it defaults to not applicable. We only put that in for like spontaneous and ENU where that's really important and where that's usually part of the assay. Germline transmission, we only do for things that come out of ES cells. So all of the old TMs, the targeted mutations that were created in mouse embryonic stem cells, those have transmission records because we have a lot of things, especially from the knockout mouse projects that are still in cell lines. And so we want the users to know, oh, this hasn't actually been transmitted into a mouse. So if you try to go get this thing, it's a cell line. Collection is for large collections of like, all the lexicon, all the alleles that were made by lexicon, the delta jon. So that's also not applicable to this. It just stays as not specified. Mixed is for cell lines. We don't fill that in. So a lot of this stuff just defaults. So then we have the things we're going to have to go back to the paper for. So up here, we have a whole bunch of buttons that open note types. If there is an existing note, it will be green like this. The marker detail clip is actually a description of the phenotypic effect of this mutation. And that applies across all alleles for this gene. And it's there so that if you're doing full curation, you know you have to update it. But we don't have to worry about that one. We do have to worry about the molecular note. For the molecular note, we've got to go back to the paper. And honestly, in this case, it would be super easy to just look at this, but I'm going to go look at their methods because I want to see if they give us more details. So basically what we want to do here is just describe what is the change in the genome for this particular allele. And what they've told us is that they target it with CRISPR and they targeted exon 1, and they don't really say precisely here what the region is that was knocked out. Yeah, they give you the guide RNAs, which is okay. We can add those in. So we can grab that. So I typically, when I write these, because there are multiple endonuclease mediated techniques, I usually start mine with just CRISPR Cas9 mediated. Just because it can be helpful for users to know what the guide RNAs were.
Mhm.
But then what I would like to see is, can I get, so guide RNAs targeting exon 1. Question in the paper is there a better, more detailed description of exactly what deletion was created? Yes, they targeted exon 1, and from that picture we can see they didn't target all of exon 1, but is there information about what they took out? If we scroll, let's see, there was a picture somewhere. All right, so the first half of this paper is about the delta exon 2 one, and then we get to delta exon 2. Yeah, this is the part. Okay, so here we go. Now they're saying they showed deletion of the rear part of exon 1 and partial insertion of intron. Okay, well, that's a little... Okay, so they targeted the CDS region of exon 1 and the contiguous intron region. So let's grab that. Targeting.
And so most people who are going to be looking at this record they're going to look at that and understand, okay, I know what the implications of this are. So for example, I don't know what CDS stands for.
It's the coding region. Yes. So that's the coding region That also, I will often do this, is Control F CDS
Hmm.
and
Protein coding sequence.
protein coding sequence. So yeah, that's
That's
a
a
key
terrible
thing.
acronym.
Yes, because it doesn't it's like CDS, it's coding sequence. Okay. And and so then they say deletion of the rear part of exon 1 and partial insertion of intron. And then they say look at figure 6C because that's a little confusing as to what they mean. I think that means that probably they got run on. So they created a deletion that removes the rear part of exon 1 and some part of the intron, and probably in read through you get some bleed through, but we're just going to take that as resulting in deletion of the exon. I'm just going to leave it there because that's the important part. The other thing to look at here is, did they say anything? Oops, we're too far down now. About, yes. And then they tell me that the RNA RT-PCR did not detect. And what I want to know is, did they confirm that this is a full deletion, that there's no expression? Yes. And so they tell me that immunohistochemistry confirmed absence of expression in the whole brain. So we'll also capture that and we'll fix the copy issue. Okay. So that gives us a nice description that says, all right, here's what was changed, and here's what we know about the effect of that change. Key things. We then need to capture the strain of origin. We don't worry about the parent cell line or cell line strain because they didn't do this in a cell line. They did this in a mouse. In this case, this is not a great reporting of that. So we get to the methods. We look at the methods. Why am I? Oh, that's because I didn't go far enough. I do not love that the methods are after everything. So they say male black 6 mice were used for all experiments. Okay. I guess so this is where you have to start deciding, are you confident that they mean that they use them here? I mean, because I've already have doubts, because there is no such thing as a black 6 mouse. A black 6 is a strain family. So there are black 6J mice and black 6N mice and black 6NJ mice, but there is no mouse that is just black 6. So I already have some ambiguity here. That's okay. And then I have to decide, well, they were used for all experiments. Does that mean they were used for your targeting? They say that it was custom gene edited service at this company. I usually then end up Googling, like, what's this company and what exactly do they do? Which is not, I think, the best of companies to look up. All right, so we've toolgen, it's English. We can sort of look for, yeah, so they do CRISPR-Cas9. I spend a lot of time going to websites in languages that I can't read because so many of the targeting services are in China and Japan, and I'm like, or Korea in this case. I'm like, I don't know what you're doing. So they have this, and so they talk about their editing services, because if you've had a company do it, then do you actually know, like, did you specify? So this website does not give me a lot of information about how they do their targeting. So then the other thing I can look at is up here where they talk about the mice. And this is kind of what tipped me in this case, was in the mice section. They call them B6, TWIC1, new KO. I
The
have a
B6
little
was the black 6. That's like what
B6
we
is
were looking at
black
earlier.
6. They also say they got their B6 from Orient Bio. So now they've given me B6 CRL Ori, but that's still, I could probably go look that up. But they also then say B6 TWIC1, and that's not quite right, because the other allele was made on 129 and not B6. But I think the preponderance of evidence made me decide that it's probably okay to say that this was made in black 6. So I'm going to put that in instead of not specified. We do have the option to just say it wasn't specified and we don't know. We also, obviously we can put in things that are not actually a mouse, but are at least get you into the strain family so that you have some idea. And I tab out, and as long as I haven't made a typo, this thing will go, okay, yeah, that's an
So
existing
it's
strain.
checking it against a controlled vocabulary in the
It's
background.
checking it against our strain table. So there is a whole nother module up here, which is a strain module. And so if I had typed something incorrect, and I mean, I can do that. I can put a G on the end of that, and that'll definitely be wrong. and I will get a little pop-up that says does not exist.
Nice.
I can either say, oh no, I really do need, I meant what I said there. And if I click OK, it will add it to our strain table. And I can also go, oops, no, I made a typo, and I can cancel and remove that. So yeah, that's a handy like support for curation. Synonyms are key.
By the way, just a question, like what percentage are we through this curation so far?
We're almost done.
Okay, cool.
We're actually pretty close to being done. So we try to capture synonyms that are informative. If they had just called it gene, synonym KO or gene synonym minus, I wouldn't bother to capture that because we already captured the gene synonym, and everybody calls things KO or something, but they called it NKO to distinguish it from the exon 2 one. So I'm inclined to take that because it's probable in the future that if somebody refers to this and they refer to one or the other, they probably are going to use that synonym. I would enter that. That requires a reference. The other things I put in here are an attribute, and this is saying what's the functional effect of this allele.
So we already captured that with some free text earlier, but
Yeah,
now
we have free text, but then we have a controlled vocabulary that says, hey, it's a knockout. And then I can come up here and I can say it is an intragenic deletion, because it's definitely that from their description. The other part of their description kind of confused me, but at least I can say, yep, it deleted that. Yeah. So that is actually everything I have to enter. I've got a nice little box here where I can say that this was created by Toolgen Inc. And that just helps us keep track of who's making them. And in case somebody else comes back and says they got an allele from them, we have the ability to check. And this field is private, so that doesn't get shown on the main web interface. And then I can flip our status because we're all set.
Was this thing auto-saving the whole time, or is there like a we're done button?
Nope.
Oh my God, that's terrifying.
That's why you have the in progress. If I was going to step away for any reason, I could have saved it.
Okay.
There's a core set of information you have to enter before you can save, but we're all set. All these buttons are live, so I can click add, and it adds.
Ooh,
and our new
we did it. We curated.
So, and it tags it with who did it and when they did it. And you can no longer add because it's created, and it's given it an MGI ID. And if I need to change things, I can modify. If I wanted to, if I went, oops, I didn't really want to do that, I could delete it, but I'm not going to do that. Most of the time we don't, like if once it's gone public, we don't use the delete button. We actually have a deleted status so that if somebody has used that ID out in the public, we don't just throw it out.
You probably want to say why it got deleted too.
Yeah.
Maybe like a miscuration or some kind of scientific problem.
Or it turns out, oh, oops, somebody else reported the same allele, but the way it was reported, we didn't catch that it was the same allele. So that is all set. And then if we go back to our paper, just because I remember one of your questions at the end. So from the paper, we have a thing that shows you the list of alleles that are attached to the paper. And now
are we live already?
No. So this will not go live until the next MGI release. So MGI puts out new data every week, but what happens is they take everything that's in the database on Monday, and then they process it for several days, and they do loads and all sorts of things. And so the data that's in the database on a Monday will come out on the next Monday. So this will be a little over a week before it is live. And that's just because the schema set up for the back end is very much like everything is in a single spot so that you can curate efficiently. The front end has a very different schema so that you can search efficiently. And it takes a lot of time to chug things along from one format to the other, and bring in all the outside data that gets merged in. But we do have on the production interface a way to go check what it will look like when it is live. The only difference between this and the live is that like our nomenclature notes, which are private, won't go out to the live database, but I can see everything that's here. So, if I open up the other one, I got to reopen this because I clicked it. All right. So this was the one that was existing. And so it has a page like this, but if I click on this, then you can see. So basically what we've done here is filled in a lot of the top part and this section and then this section. The rest of this comes from other annotations, and then the one that we indexed will eventually show up in the all reference list. But this display lets me look at all of this and say, okay, yep, this all looks good. It's helpful then to look at the notes and try to see, did I make any typos? Are there any weird things going on with the display? Like I noticed there's an extra space here. So I would like to fix that because that looks a little weird that there's an extra space. So I can do that and I can modify.
For those who are watching, this is one of the core skills of a biocurator is to recognize incorrect spacing.
Yeah. So then that is fixed and that is instantaneous. So the relation between our editorial interface and this back-end production interface is immediate. It doesn't make it to the public for about a week. The fastest it would make it is a week. So it's just
amazing. So that's it. That's the whole story from beginning to end. Minus we cut out a little bit of the pre-planning and pre-reading. Yeah. So I think we're kind of at the end of what you wanted to show, right?
Yeah. Yeah. So
all
I can
right, then I would suggest unscreen sharing, and we should have a couple remarks at the end, and then we can close out. So we spent a little bit less than 40 minutes going through that. Originally, the plan was to see what we could do in 10 minutes. And I think, well, that was an ambitious plan. So do you think that was medium, an easy, or a difficult paper to curate?
That was an easy paper.
So how many of these do you think you can manage like in one day? Are you going to say, I have time for 4 of these? And like, that's your work day?
Well, yeah. So it is working them in between meetings. So an easy, like this took me way longer walking through it with you than it would normally take me. Like I was thinking, oh, we could do this in 10 minutes or 15 minutes, maybe a half an hour. So you can run through, but it's a bit of the pre-planning, like the pre-reading, but usually to like start to end, enter the allele is under a half an hour.
Okay.
The figuring out the new allele and getting everything set can take a little longer than that. And a bad paper will take a lot longer.
All right. So follow-up question on that. What would you like authors of these papers to do to make your life easier? And we already kind of mentioned a couple of those things, the way that the information was organized, but like final thoughts on that.
I mean, if they really wanted to make our lives easier, they would pre-submit.
Mm-hmm.
The more you can pre-submit, because like I just made up a lab code for this person. If they had pre-submitted, they could have picked a lab code from those available. Like, for people out there who are listening, if you've ever gotten a weird email from ILAR that says, oh, your lab code has been registered. Yeah, it's us. Or maybe it's RGD, the Rat Genome Group. Because we can't afford to wait to like ask people what code they want, because that would take forever. So we just create them for you. So if you pre-registered, then you have some control. But yeah, the more like if you can submit the data ahead of time and the more you can use IDs. So like the papers I like have, all of the other alleles will have their IDs in there. Like these guys gave us a reference. They could have given us the MGI ID for that allele. And that would have been great. And then, some of the AI tools that we've been playing with to try to speed up the indexing steps, the steps of like just identifying what's in here, would have an ID to work from and could go a lot faster.
Okay, so you headed me off because one of my follow-up questions was, do you think that where and how does AI have a place in the curation and the maintenance of your resource going forward? And so using it to do some of the busy work ahead of time is a really good explanation that a lot of people have. But is there anything else that you'd say about that?
Yeah, I mean, that's a thing that the Alliance is actively working on. So we have like when I showed you the reference module where, we have machine that goes out and grab things and it does like the machine learning search and scoring to tell us, oh, these are the papers that are likely to be good, that likely have information we want. So that's great. And that doesn't involve AI. It involves, sort of, or a different type of AI because the machine learning side.
Mm-hmm. Not LLMs
Not
and
LLM
ChatGPT,
side.
different kind of AI.
But we're trying the LLMs in the Alliance for that indexing step. At least that's what we've been looking at so far is, hey, what are the alleles in this paper? Can you figure out, go look in the database and bring me back the IDs? And we've been having some success. I mean, it's definitely rocky, but it's getting better.
Okay, so we're back from some minor technical difficulties. And the last thing we were talking about was the usage of AI, both sort of typical classic machine learning and then sort of the new idea of AI, which most people mean LLMs when they're saying AI these days, and ChatGPT and language models. So how do you think that those are going to fit into the maintenance and the curation of MGI going forwards?
So not so much on the MGI side, but on the Alliance side, as we move there. The team there has been developing a custom LM based model. So some other folks have been doing more of the testing. And one of my colleagues is like, Michelle has been comparing what they're developing with just using the off the shelf. And what we've got going is, she said, miles better than the off the shelf, because what LLMs really need is a lot of guardrails. It is a lot of control to say you can't make anything up. And here are the hints and tips that the curators know for, like, oh, if you're looking for the MGI allele, well, we tend to use the author name in the lab code. And so if you know that and they mention a name, you can go look to see, oh, is there an existing allele in MGI that has the right type that has that name? So I'm trying not to be overly pessimistic, because I
You're in
tend
good company.
yeah, I tend to be a bit on the AI side, but I mean, I think I can see where it might be very helpful at extracting. Here are all the alleles that they've hidden throughout the paper, and here's what we think they are. And now you can double check and confirm and put them in. There is no intention so far of taking the human out of that loop, of not having somebody confirm that this is actually correct and that this is the right allele. And right now they've got it so that it won't. One of the early tests I did, I was like, well, it's just giving me one and it's giving me the wrong one. And that's not helpful. So now it's gotten better at saying, well, I don't really know, but here's 3 it might be. So you can go check that and see, oh, well, that one's actually the right one. And so that can be helpful, especially for the papers where in certain fields, mice just appear in the middle of the paper with no reference in the methods, which is a thing I would love if they would stop doing.
Hmm.
So then it's useful, because if you've got to comb every figure caption to see if you found every allele, you're going to miss some. So if you can have something else comb first and you confirm that, that should probably be helpful. So
Great.
I'm
So
cautiously optimistic, I'll say.
good. And it's not just your group, Alliance and MGO, that's interested in this, but many members of the biocuration community are doing this. And you and I, as members of the ISB's executive committee, have helped in the training and outreach committee of the ISB. We are planning on February 9th an AI in biocuration workshop where people are going to do a little bit of show and tell on the technologies they're developing in this. So anyone who's watching this who finds that part interesting, I would suggest to attend that. And we'll have a link to that wherever this video ends up. And our
Right.
ISB channels, whether that's on Mastodon, Blue Sky, or our blog and LinkedIn, we're posting links to that so people can put it into their calendar and join us virtually on Zoom for free, like most of the events that we host in the ISB.
Well, and one of the speakers at that is Chris Tabone, who is the one who's developing the Alliance AI tool.
Great. And I have one final question, which is also a big one. Sorry, this isn't an easy question. What's the funding situation like for your group and more general?
Yeah, you say that's an easy question. That's a question I ask my supervisor periodically. It is a bit in limbo right now. So MGI has multiple groups. MGD is one of them, and that's the one that funds most of my work. And we got a really great score, and we're still waiting because funding out of NIH is very, very slow because of the staffing cuts. And I mean, the funding situation has been getting tighter and tighter and tighter while I've been here. I mean, I started more than 20 years ago, and we used to send curators to, well, there used to be more curators, and there used to be more software engineers, and we used to have money to travel. And now there are fewer curators and fewer software engineers, and we don't really have any money to travel. So things are tight, but they're tight for everybody.
I just realized this was a super negative question to end on. We should have
Yes,
ended with
yeah,
something
because
else.
it's not a happy question.
Despite all of the issues in funding, I think people in our community are adapting and finding ways to get the
Yeah.
job done and to build up resilience towards funding cuts and towards, different groups having priorities in different times. This is something we're all thinking about.
I was gonna say, and I'm talking about the NIH, the big grant, but the PIs, so the primary investigators whose job it is to get the money, they don't just look for one funding source, right? They're constantly looking for new ways to fund things, and other sources of revenue and other ways that we can keep going. I mean, I think one of the problems we all run into though is a lot of the funders want something new, and yet we also need to maintain and update. And so there's always trying to balance that. But I mean, I think to end it on more of an upside, I did say I've been here over 20 years. So, it is a career that you can have longevity in. It is reasonably stable, knocking on all the wood around me. So, that there is that. There is, I think, a positive note is that it's a good career. It's one I've really enjoyed.
I think that's the perfect place to end it. So thank you, Sue Bello from MGI and Alliance and Jax for being our inaugural interviewee. I hope that this is interesting for people to follow along and to see how the curation is done. The
Yep.
goal is we're going to post this video, maybe cut up a little bit to get rid of our technical difficulties, along with some links to some of the things that we did. So I think that we need to make sure that we have a link to the thing that you curated, since it's got an MGI ID, and by next week it will be published. We probably
No, the week
next
after.
2 weeks. 2 weeks. Okay, in 2 weeks it'll be published. We can link to some of the most recent papers from about MGI and from MGI. Probably should also link to the paper we curated today, just in case people want to follow up on it.
Yes.
And I would also think we should link to some of the other interesting things coming up in ISB, like this AI and biocuration workshop, and just like our social media for the International Society for Biocuration. We'd love for people to get involved who haven't already been involved, even if they're not within the bio world. The ISB is also servicing people in the chemistry and even bigger within the sort of natural life sciences sphere. And even some people outside of these natural life sciences are interested in the sort of ways that we talk about things and do things.
Yeah, and the way we standardize and I think, yeah, it's good for the various groups to interact to see, oh, well, yeah, that innovation you've made would really be helpful over here, even if I'm not applying it exactly the same way.
Great. I think we're done. Thank you so much, Sue. We will be in touch soon. Thanks everybody for listening. Bye bye.
Right.